All right, great. Yeah, so still Devin Lange. And for my second talk today, I will be discussing work that we submitted to IEEE Viz this year, hoping to hear back tomorrow on that. And this project is going to look at single cell microscopy again, totally unrelated project, by the way, but now for live cell imaging. And this project is a collaboration with two labs at the University of Utah that, broadly speaking, do research to understand cancer cell development. And the lab that I'm a part of, the Visualization Design Lab, again, broadly speaking, develops visualizations for groups like this to help them analyze and understand their data. And this understanding cancer cell development is quite broad, but they do this with like fairly similar technology for a lot of their questions. And this is an automated microscope setup where they can watch cells grow over time and collect information on them. And the types of questions that they ask are, how do healthy cells turn into cancer cells? How do those cancer cells spread throughout your body? And how can someone who's been diagnosed with cancer, can we look at their cells and develop an effective treatment plan for them? So this last question in particular, right? This is sort of like the precision medicine. A cancer patient goes into a hospital, you do a biopsy with their cancer cells, collect data with this microscope, and based on that data, develop this plan. And this idea was the major focus for the first visualization system that we developed for them. We published this 3 years ago now in Viz 2021, and actually presented at BioVis my first time virtually that year. And the work today is an extension of that collaboration. Yeah, so this is the previous work, Loon. That's lots of nice visualization views for doing this comparison across like drugs and concentrations for like cancer cells. And today's work is, as I said, an extension of this. And the main idea of this is, how can we use this visualization specifically for this problem of complex multimodal data? So to begin, I'm gonna talk a little bit about what I mean with this multimodal data for our collaborators in particular. So as I've already alluded, they do live cell imaging, so images of cells as they grow and divide over time. They run some processes that track those individual cells, so look from frame to frame, see how this individual cell is growing and developing. One way to do that is to characterize that with derived attributes. So at each moment in time for that cell, calculate things like the size of that cell, the shape of that cell. It could be like the amount of a specific protein. And since we're recording these attributes over time, what you end up with are these time series trends. And then as you go forward, cells divide into two daughter cells typically. And if you record that point of division, what you end up with is this tree of cell relationships. So taking a step back, abstractly, the data that we are working with here are these stacks of images, this time series data, and this tree data. So these are the three data modalities that I will be discussing in this talk. And to make this a little bit more specific, I want to talk about some examples. So let's say we have some time series data of a cell. That cell grows larger, divides into two daughter cells, which grow, then divide into 4 cells. Plotting this information on a line chart is very informative. There's a lot you can learn from this. But what if your data looks like this, right? In this case, we're seeing a cell that is growing really abnormally large compared to all the others. In this case, is this a cell that is actually growing that big in reality? Or is this an error or flaw in your data processing? The only way to answer that question is to find the corresponding cell in your image stack and look at it, see what your segmentation is, see if it looks really large. In this example, we see an interesting pattern in the line chart, this divergence of growth where some are growing quickly and some are not growing as quickly. And a question you may have while looking at this is that fast-growing cell in the second generation propagating into the fast-growing cells in the third generation? Again, you can't determine this just by looking at the line chart. To answer this question, you now need to look at your other modality of data. You need to look at the tree information here. And really the point of these two explanations is, at least from our experience, our collaborators are constantly looking across these data modalities, linking these data elements together to try and understand a full picture of what's going on with the data. And this is kind of hard a lot of times. I think Eric mentioned in his talk that his collaborators have this case with 3D data and then Excel files. We experience the same thing. Look up, find the exact ID, look in this other place, look through the things, find the ID, look back and forth. So really the goal with this project is to create a more integrated system where we have these visualizations, we call them composite visualizations, that are displaying these different data modalities together to make it easier for the analyst. And of course, we don't just like smash them together like this. So let me talk through how we actually construct these with an example. So the first example will use tree as our primary data source. So for all these, we select a primary data source, which serves as our host visualization. In this case, we have this sort of node link diagram for our tree information. Then our secondary data types we can embed as client visualizations. So here I am nesting the time series data within the nodes of the tree. Similarly, you can superimpose data like the images alongside those time series data points. You can do this automatically, or you can do this on demand as the user is interacting with the visualization. So let me take a minute here and switch to our actual system and demonstrate what this looks like for us. So what you're seeing here are 3 composite visualizations. To begin, I'm going to look at just this one on the left-hand side. This is our tree first visualization. And if I expand this, we can see the time series in these colorful ribbons, and then the images are automatically placed at key points. So this could be like where the cell divides, or it can be where those attributes are changing very quickly in a short period of time. Now, as I expand this view, more images are pulled in automatically. And similarly, as I condense it, we only show what we have space for. I can mouse over this time series to really select any cell along this point that I want. And selecting one of the auto-populated thumbnails will pull in the previous and next frame to that image. And here, actually, we see something quite interesting. If you look at the images, what you see is the cell is actually dividing in reality, right? Between these 2 frames, that cell has divided into 2 daughter cells. But the tracking algorithm used here has decided that that one daughter cell is the same as the parent cell. So this is an error or flaw in this process that we can see here. Great, I'm going to move on to our second composite visualization view. In this view, the time series data is our primary data type, which we can embed, which we can plot as a line chart here. And as the user selects one of these lines, we can interactively show the relationships for that particular cell. So we don't show every single relationship, but we've selected this cell, and the green dashed line is showing me the parent cell and the 2 daughter cells of the one that I have selected. Similarly, on demand, you can show images of the cell at that corresponding point in time. So jumping back to the visualization system, let's look at this in action. Here we have our line charts. And as I mouse over, you can see the images of that cell at that moment in time. And when I select this cell, we can see those relationships. So again, stepping through this, the yellow line I have just selected is the currently selected cell, right? And I think you can see this on the monitor. That first dashed line is showing the parent cell to the selected one. And then moving along, there are 2 dashed lines to connect to the 2 daughter cells. And the daughter cells are assigned different colors, green and purple. And these are just there to help you distinguish them. And those colors, green and purple, are continued to the final generation we see here. So the green cell divides into 2 green daughter cells, purple, 2 green daughter cells. And again, we see sort of an interesting attribute in the data here, which is that divergence in growth. So these green and purple cells, remember, all came from that one cell we selected, but we see this divergence in development of these cells. Great, so for paying attention, there should be one chart left. Here we are looking at the imaging data as our third primary data type. And encoding this is fairly obvious. We have images, we can show the images directly. And the time series data that we are superimposing here is the past positions of the cell. So this is essentially cell movement from the past. And the cell relationships are shown in green. So we have green marks for the current cells in the image, and then lines drawn between them connect the siblings with intermediate marks, intermediate circles indicating parents and grandparents. So within the tool, this is what it looks like when one cell divides into 2, then 4, then 8. And in this experiment, we actually reach a point where the cells become too populated, too dense, and they actually start to overlap. So this image view, it's easier to get the spatial relationships and identify things like this. So as a recap, we talked through our approach to designing these composite visualization views. We talked through 3 examples. And I like this work because there's sort of 2 main contributions. The first is sort of this visualization design theory, like how can we think about these visualizations and try to create these nice composite visualizations. But we also have this real tool that instantiated this work that is usable. And on the note of this second piece, this is an ongoing project. This includes not just this lineage work, but the previous paper that I talked about earlier. And we are working on this, making this a more robust tool. And yeah, basically, if you work with live cell imaging data and are interested in any of the things I've talked about today, feel free to reach out to me directly. And yeah, we can maybe discuss trying to get your data into the system like this. So with that, I would like to thank you all for having me give this talk. And yeah, happy to answer any questions. Thank you.
I don't work with cancer, but I do work with cells that I'm interested in watching how they, I'm a microbiologist, so I'm interested in watching how cells, live cells that are dividing and interacting, do they have to, like cancer, only divide? Like, is there any kind of flexibility in your tool for like cells to maybe like disappear or have died, or like, is it only just increasingly multiplying, like the tree goes from few cells to multiple cells? Like, is there only one way to go?
Yeah, so the question is like, do the cells have to divide? And I guess there's sort of 2 answers. The short answer is like the tool will still work even if the cells don't divide, right? The vis will just end. But like the deeper point of like, what about cells that die or have these other things happen apart from dividing? And I think that was something we were interested in encoding directly, right? It might be interesting to say, these are the cells that died. But at least for the data we work with, like this, it's like a challenging data set to acquire, especially at scale, right? Because cells move out of frame, cells overlap, and these cause tracking issues that make it very difficult to distinguish like cells dying versus these other things. Like it's possible to a certain extent, but we just haven't done it for this project.
Sure. Like, but it would work. Like, they are dividing, but like it could also accompany. Like, it could just stop the tree branches if the cell disappears or something like that.
Yes, okay. It will work. Even if you do no tracking at all, technically the tool will still work. Your trees are just all going to be just the one line.
Sure. Yes. Yeah. All right. Well, thank you so much.
Sure. Yep.
I have a quick question. Thank you for your talk. So if we're doing the wood healing experiment, so we want to look at the cell usually move faster at the edge of the, I don't know whether you understand the wood healing experiment. Once you have a convoluted cell, we just destroy a cell with something, and you have an empty like road, and the cell will move into it. So how you can catch up? Can you catch up speed, and also the how cell divide and also move.
Yeah, so what I heard is like for different experiments like this wound healing experiments, like what are the attributes you can measure in this tool? And I'd say this is like pretty flexible. And currently some of the processing and visualization is a little bit separate. So whatever you encode in your data, the tool can load. So it's already expecting like positions of cells, but if you want to like add derived attributes of like the cell's instantaneous velocity or cells like total movement covered, like some of our data sets actually have these exactly already. And once you have those, you can visualize them within the tool. Yeah.